We have a first speaker, Dr. Sean Collins from UK Health Security Agency. Dr. Collins is biosafety research scientist in the Biosafety Air and Water Microbiology Group at the UK Health Security Agency. He has been a part of the group for over 2 years. He has worked on many projects while working for the biosafety group. This includes environmental microbiology, decontamination, aerobiology, water microbiology, as well as virus work, which includes helping carry out diagnostics in the monkeypox outbreak response. He was also involved in investigating how different viruses such as mpox and SARS-CoV-2 persist as aerosol in the environment. He has taken part in air sampler validation work to ensure when these samples are taken on sampling trips that the use of the equipment is optimized. He has also processed and analyzed samples taken from different environments such as hospitals and workplaces. Thank you so much. With your varied and expert background, you are starting this session. Welcome.
Hello, hope you can all hear me. I'm Sean Collins. I work for, as introduced, the UK Health Security Agency. You may have seen me earlier presenting a poster, but maybe not. It's a different topic anyway. So I'm going to be talking about the validation of air sampling for SARS-CoV-2 and mpox using the Sartorius MD8 airport air sampler. Just before I start, you may not realize that mpox used to be called monkeypox, but last year it's changed its name. So just in case you're wondering what I was talking about. So a bit of background. So during both SARS-CoV-2 and mpox outbreaks, a lot of sampling took place in hospitals and other environments like workplace environments. And part of that sampling process, once we knew that SARS was transferred, could be aerosolized, air samples became important in both these outbreaks. So because they were outbreaks, a lot of samplers just got used. Not much could be done in terms of validating because it was an emergency. So this is why we did some of this work. So 2 examples of air samplers that were used during the outbreak and during some of our sampling trips were the MD8 airport sampler, which I'll be talking about today, and the Carolus Micro air sampler. So the Carolus air samplers are the bottom and the MD8s are the top. So this both slightly different in the way they work. So Carolus Micro, which has said the one on the bottom, you can see the plastic thing attached to it. So that usually contains about 15 milliliters of liquid of media. So the air gets drawn through into that liquid and creates a cyclone, and the particles get stuck in that. Whereas the MD8 draws the air through a gelatin filter, which you can see placed next to it on this slide. And then that is then recovered and taken off, ready to be processed. So during both outbreaks, the viral RNA and DNA was detected in air samples collected from patient rooms and other contaminated environments using some of these samplers. So for the MD8 airport, some references there. They found CT values about 41 and 38 in one paper, but no, they couldn't isolate any viable virus. And that seems to be a common theme for both recoveries. So a potential contributing factor is that the time between collecting and processing air samples can be more than 24 hours. This means that transport and storage conditions could influence the recovery of viable virus. because in ideal conditions, as soon as you take these samples, you'd be processing them, and but this obviously can't happen if you're going to someone's workplace or to the hospital. This needs to be taken back to the lab, and this transport and storage could be at room temperature, 4 degrees in a fridge potentially, or on ice, or in a -80. However, -80 or on ice could cause an unwanted freeze-thaw cycle, and all this could affect the virus viability. So just a bit of back more on the air sampler. So the MD8 air sampler, as I said, pulls air through a gelatin membrane filter, which is then dissolved in warm virus media. We say warm because it's the only thing that gives us an advantage to manage to actually dissolve these filters. It can be a bit of a pain. However, so in this study, gelatin filters were contaminated with SARS-CoV-2 or mpox to investigate the following aims. So determine the effect of storage or transport conditions on virus viability and recovery, and also the impact of sampling on virus viability. So the pictures at the bottom are just some of the equipment that we used in this study. So the bottom left is a flexible film isolator, or FFI. So this is used for because we're using containment level 3 organisms in SARS and mpox. So this is containment that we work in when you doing any aerosol stuff, any aerosol work. The second picture is the spray tube that we spray the virus through, and then the next picture along on the right is the actual filters that we impacted and contaminated with these viruses. So before we did any cell-free work, we had to determine some conditions that we would be using. So we did some pre-work at containment level 2. So these earlier investigations involved the use of MS2 as a surrogate virus to optimize system test conditions. And so this involved duration of spray and filter loading capacity. The reason we do this is because it allows us to work more easily and look at more conditions and work more safely. Because part of the problem we had was we had to, as I showed the picture on the previous slide of what the sampler looks like, which you attach this filter onto the end of the sampler, we needed to be able to impact the filter directly through the SARS or mpox that we're spraying up. So the picture on the bottom left is called a bioair machine, which can control airflow. It also can control humidity, and it keeps track of temperature and how much air is flowing through each of the colocin nebulizers, which we're using to spray up the MS2 in this study, in this part. So one colocin nebulizer, 6 jet, spraying water to maintain humidity and flush the system, while the other one is spraying MS2. So the middle picture is how we ended up attaching the tubing to the gelatin filters. So we were looking at 1-minute sprays, 5-minute sprays, and 10-minute sprays of MS2. We ended up going with 5-minute sprays at 50 liters per minute. The reason was because the 1-minute spray we didn't get as much recovery as the other two, and the difference between 5 minutes and 10 minutes wasn't worth using up more stock and spraying for longer. So this is what we did for our methods. So the top left picture, as I said, was a flexible film isolator. So the machine you can see in the bottom left of that picture is the bioair that we use for CL3. So it passes, so it controls the spraying that's going on this time in containment. So from the outside, you can control how much the nebulizers are spraying, or make sure there's no leaks in the system, because it keeps track of the pressure that you can alter. And then there's 2 people who can work in there together. And then, so So we directly contaminated these filters, gelatin filters with SARS-CoV-2. We needed high titers so that we could see if there was any difference in viability. So we used 1.3 times 10 to the 6 PFU of SARS. We used the England 2, which was the first SARS that was isolated in the UK. And we had high stocks and high titers of that, which is why we chose to use that. And then mpox was a similarly high titer at 3.5 times 6 PFU. So we had 12 contaminated filters, so split into 4 sets. Each set was in triplicate. So set A was the ideal conditions, as I mentioned earlier, but this isn't necessarily what you do in the field. So that one was processed in 2 hours and plaque assayed. And then set B had the additional freeze-thaw cycle in there. So this was plaque assayed on day 2. Set C was as if you stored it in a fridge overnight before plaque assaying, and set D had an extra freeze-thaw cycle to what set C had. Okay. So this was the second name, so the second part of the methods that we were looking at. So these filters were also used to study the effect of the air sampling process on virus viability. So if you are sampling and the filter is picking up any virus, and you continue to use the machine, the air isn't damaging the viability. So following contamination, test filters were attached to the sampler using the same process as shown in the previous slide. Same equipment before attaching it to the sampler. So we did volumes of 250 liters to 2,000 liters, as shown on the right. We did three replicates. And this was to assess the potential inactivation of different sampling strategies. And then afterwards, these were processed immediately. So for the first part, this is the SARS results. As you can see, the set B caused a 0.31 log reduction, which was, so that's the freeze-thaw cycle. That caused a significant difference compared to A. So set A, because this is the ideal conditions, all the other sets are compared to this. So set C, which was being stored in the fridge overnight, caused a 0.69 log reduction. And then the actual freeze-thaw cycle after storing in the fridge caused a 1.5 reduction, and all of these were significant. So the mpox results were slightly different. So the investigation into why they had different results is still ongoing, despite them both having similar titers. As you can see, the freeze-thaw cycle seemed to give around the same, didn't seem to make too much difference, but there was no significance in that. But storing in the fridge gave the biggest loss of virus. Again, this was compared to set A. So this is still ongoing. And then the second part of the study showed there was no observable difference witnessed in viral viability between controls and filters exposed to the air sampler. So there's no significant difference between any of these results on this graph on the right. And this is probably expected because SARS-CoV-2 is a pathogen transmitted by aerosols, which explains it being stable as an aerosol in this part of the study. So, in conclusion, immediate processing of filters of SARS-CoV-2 resulted in greater virus recovery with less than 1 reduction in viability observed following overnight storage. So mpox, as you've seen, gave different results. The further studies are needed to understand why this difference has occurred. And as most real-world air samples collected will require transporting from your hospitals or your workplaces or any sampling location to a lab for processing, it is imperative that users fully validate whatever sampling methods they identify for recovery. So then the duration of air sampling that we observed did not appear to affect the recovery of viable virus, with similar SARS titers identified no matter how much time you sampled and used the machine. So it's likely that the gelatin matrix, these filters, could be what's protecting the virus particles from desiccating of effects of prolonged sampling. The virus is also transmitted by aerosol, so it's likely to have resistance to desiccation. So future work, as I keep saying, repeating the investigation with mpox is required to see whether there's any part of our methods that this the reason for these results. Investigations using a wider range of biological agents to determine most appropriate transport and processing conditions, and then also new air samplers which we need to validate, such as the Sartorius compact air sampler, which is slightly different to the micro that was mentioned earlier. The micro uses, as I said, liquid to recover into, whereas this draws the air into that similar cone, but it's dry, so it sticks to the edges, and you just rinse it out, which would be easier. So it's easier to recover than the Sartorius MD8 sampler, because as I said earlier, the gelatin filters can be quite difficult to actually dissolve in media, and as much as we heat the media, because working in CL3 cabinets, it cools quite quickly. So yeah. So some acknowledgements. Thanks, Tom. Good luck. Any questions?
We welcome questions from our virtual participants in this session. Please put your questions in chat, and we request the participants here, when you come to pose the question, please state your name and affiliation. Thank you. Yes, sir.
Nice presentation, Sean. Worth Calfee, US EPA. I was wondering if you could comment on the utility of air sampling for mpox. Is it useful for estimating exposure risks, knowing how the virus sheds or contamination spreads?
Yeah, so we, go on, Tom. No, yeah, I was just going to say that during some of our mpox sampling trips, we did manage to, in patient rooms of mpox patients, we did manage to recover with air samplers viable virus. So I would say that it is useful.
It would spoil half my talk. So yeah, I'm going to deal with it in a bit later on as well.
Fair enough.
Yeah, Catherine Ratliff, EPA. So you looked at changing the sampling time, but I was wondering if you thought about changing the sampling flow rate. 50 liters per minute is relatively high, I think, for an air sampler. Maybe it's not something you could change with that sampler, but I was curious if that's something you thought about for optimizing recoveries.
Yeah, so I think we use 50 liters per minute. Oh my God, I think we use 50 liters per minute because that's just the go-to for using that the bio machine. And I think all sampling I think all groups use 50 liters per minute.
Thanks.
I'm Randhir Makkar with the Guild Biosciences here. I have just one doubt. When you were contaminating your filters, were you spraying the virus on the filters, or you were just putting it and then putting the airflow?
Oh, sorry. Maybe I should explain that better.
Okay.
Yeah. So so we were spraying from a colocin through a tube straight onto the filter.
Okay.
So we attached it straight to the tube. So it was a direct contamination of the spray.
Yeah. Thank you.
All right.
Hi. So really really good presentation. Question on the humidity. When you did the sampling and you let the 50 liters a minute flow, do you happen to know what the relative humidity was? Because we've seen some things where dehydration can be a problem with the spores, or of the virus, but also when you over humidify aerosols, that also affects viability.
Okay, good question. So in other studies we've and using similar equipment, we've either done like low, medium, and high humidities. On this one, we weren't trying to set a humidity, so it was relatively low until the spray happened, and then obviously because the spray is shot up. But we didn't try and reduce it; we just let it go. So I'd say it's about 40–60% during the spray. So slightly medium relative humidity. All right.
All right, thank you.
Thank you so much.